Hi, my name is Fernanda, and I'm going to talk about some topics of genetics for my biology project. So, let's start. Let's talk first about chromosomes. What are chromosomes? Well, chromosomes are the structures where we're going to find the DNA coded and packaged. A chromosome is composed by two chromatids, 1 and 2, and we're going to find chromosomes in the cell nucleus. The number of chromosomes in a nucleus cell is going to depend on the animal and species. In humans, we are going to find 46 chromosomes. If we want to see closer the DNA, it's going to look like this. The DNA is a deoxyribonucleic acid, and is where we're going to find all the information about us. Each DNA in each person or animal is unique. There is no same DNA. Now I'm going to talk about Gregor Mendel. Gregor Mendel was an Austrian monk that discovered a lot of basic concepts for genetics. The basic concepts about heredity. Now I'm going to explain Mendel experiments that he doing observations in his garden. Pea plants were the flowers that Mendel used in his experiments. He cross-fertilized two different flowers with two completely different characteristics. For example, one purple, one white, one yellow seed color, and one green seed color. So, after observing and analyzing the results, he became with great ideas about heredity, some laws and some theory that I'm going to explain now. The first and basic thing is about characteristics. There is a big difference between characteristic and trait. Characteristic is the general thing, so the characteristic here we're going to say that is the stem height, and the trait is going to be tall or short. So here, talking about traits, there is going to be one called recessive and one called dominant. So the dominant trait is the one most common. It's the one that statistically too is going to be the majority. The recessive is the one that we're going to observe in only a few cases. Talking about the stem height, we're going to find that the tall is the dominant trait and the short is the recessive trait. So we're going to see more tall pea plants than short pea plants. Now I'm going to explain a Punnett square. A Punnett square is a chart which shows all possible gene combinations in a cross of parents. Here I crossed two different traits. The characteristic is the color of the flower. In this case, the dominant, represented by a capital letter, is purple, and the recessive, represented by a lowercase, is white. Here we can see that when we cross a recessive and dominant trait, we're going to obtain the combination of these two. That is called a heterozygous. This is going to be our first generation. How I cross it? Well, I start here seeing what letter I have up and what letter I have at the side. Here, like this, and like this. Our result was this. Now here I put our result in a Punnett square. Capital P and lower p. Our result is repeated at the side and here down. Now we're going to cross it. So here we have a P and a P capital case. So we obtain double capital case. Here we have a lower p and a capital P. So we obtain capital P and lower p. We always have to put there first the capital. So capital P and lower p. Here we observe we have another time lower p and capital P. We put first the capital, so capital P, lower p, like this. And here we obtain lower p and lower p. So lower p and lower p. That each squares represent a 25% of 100%. So we're going to look that capital P and lowercase p is repeated 2 times, and if each square is at 25%, we are going to find that lower p, capital letter P and lower p is going to be equal a 50%. This combination of a dominant trait and a recessive trait is going to be called a heterozygous. And two equal letters like two capital P's or two lowercase p is going to be called homozygous because they are equal. And our results of homozygous are going to be this. The dominant trait 25%, the recessive trait 25%, so we're going to have a 50% of homozygous. Now for the phenotype form, that is how it looks, we have to take into account that if we have a capital case and a lower case as the capital, if the dominant trait, what is going to win is the dominant trait. So we have purple and white. The flower is going to be purple because it's the dominant one. So we're going to have 1, 2, and 3 squares of the Punnett square. That, as they are 25%, they're going to form the 75%. So the 75% are going to be purple flowers, and only this one is going to be a white flower. This is the phenotype result. Now I'm going to show you how to cross two different traits. In this case, I'm going to use the flower color as the letter P and the seed color as the letter Y. We already know flower color, so the seed color is going to be dominant green, recessive yellow. Dominant capital letter Y, recessive lowercase y. So we have here, we already know that if we combine dominant and recessive trait, we're going to obtain a heterozygous of both. So I have an heterozygous flower color and heterozygous seed color. So how I'm going to locate this in the Punnett square? Well, first we're going to start with the capital letter, with the capital letter and capital letter, capital letter and capital letter. Then our capital letter P with the lower case of the other trait, like this. Then the other P with this and this with this. So lower P capital letter Y, lower P and lower Y. So this is how we're going to do it. After we locate them up, we're going to do the same down. And then we can start crossing them. So here we have lower p, no. Capital P, capital P, capital Y, capital Y. So I'm going to write it. Then here, capital P, lower Y, lower capital Y. So you only have to observe what do you have up and at the side and put it. So here up we have lower p and at the side we have capital P. So lower p, capital P. And then you have capital Y, capital Y. So capital Y, capital Y. Here we have lower p, capital P. Remember you have to put the capital first. Here we have lower y and capital Y. So we are going to put the capital first. And so on. Our final result is going to look like this. I follow the same process that I show you in all the squares. Now let's start doing the results. So we're going to do genotype and phenotype results. So here I do the genotype results. This is really easy. You only have to look for the squares that have equal traits, that have all the letters of recessive trait or all the letters of of dominant trait. So we only can find here one square of the dominant trait completely and only one square of the recessive completely. So here we do the results. 1 of 16 squares that we had here. And heterozygous, well, is all the other ones that aren't all. So that was all for today. Thanks for watching and see you later.